Thank you very much, Wenbin, and thanks to the Beckman Laser Institute for the opportunity to visit you today and speak to you today. And also thanks to all of you for taking time out of your busy days to come and see this presentation. Just by way of background, the technology I'm going to talk about today, my visit here was kind of initiated by a collaboration with Wenbin. So he's been using our technology for a couple months now. We placed an instrument in your cell culture facilities here. He's been generating some very interesting data. And so what I'll do today is I'll tell you about the technology as well, some of the work that I was involved with using this technology for drug discovery applications, but the idea is maybe to give you some ideas about how this technology may or may not be useful to your own work. And finally, I'd just like to make this a very informal talk. It's should be relatively short if I just go through the slides and you don't interrupt with questions, about 40 minutes. So please feel free at any time if something comes up, just interrupt me. I'd rather have it be an informal discussion than a lecture. So yeah, the title, Advances in Applications in Label-Free Cell-Based Assays, is kind of a very general title. Also a little bit more background about me. I'm really a biologist by training. I'm not an engineer. So I really focus on the applications. And so what I'm going to do in this talk then is introduce you briefly to the company and the technology. And then I'll provide a general overview of some of the assays that can be done with this kind of technology. Very basic categories of assays like cell proliferation, cell adhesion, cytotoxicity, and cell invasion and migration. And then kind of the second half of the talks, we'll be talking about some additional assays or some of these same assays, but putting them in the context of the drug discovery process. And this is work that I did in my previous role at ACEA, which was leading up the high-throughput screening oncology drug discovery program down in San Diego. And so in that context, we'll talk about utility of this kind of label-free technology in assay development, high-throughput screening, follow-up and secondary screening, and then finally I'll end with specific deployment of this technology that we use and pharma companies globally now use for cardio safety testing. Just very briefly, the company that I work for, ACEA Bioscience, is located in San Diego, California. It's been around since 2002. We've got a global footprint with headquarters in San Diego, manufacturing in Hangzhou, China. So in total, we've got several hundred employees. And the technology that I'm going to talk about today is what this company was built on. It's branded as Xcelligence, Real-Time Cell Analysis System. It's an impedance label-free technology. So that's what we'll be focusing on today. Right now, that technology in the form of an instrument line is sold through a number of distributors worldwide. And then the second part of our talk, when I talk about the drug discovery work, that's a separate business unit within this company, which, as I've already mentioned, is focused on oncology drug discovery, specifically lung cancer therapeutics. And just one of our marketing slides, the technology I'm going to tell you about today is deployed in a number of different instruments. They're arrayed on this timeline, starting from 2008 up until our most recent launch, which was just last year. As a side note, for 5 years between 2008 and end of 2012, this technology was distributed globally by Roche Applied Science, big provider of reagents and assay technologies. We're the inventor of this technology, so at the end of 2012, we actually took it back, and we run the commercial side, sales and marketing ourselves now out of San Diego. The reason I say that is some people may have heard of this technology, and often if they have, it's in the context of Roche. But ACEA is the inventor of the technology. Everything I'm going to talk about today with regard to the biosensor, it's an impedance biosensor, is deployed in more or less the same form in all these different instruments with a couple exceptions. So one exception here, in April of last year, we launched a benchtop flow cytometer, completely different technology. We can talk about that informally later if you're interested. This is the system that you actually have here as part of this collaboration with Wenbin. It's our lowest throughput system, and then the other instruments primarily differ from each other in terms of throughput, how many parallel wells full of cells you can measure at the same time, with a couple of specific exceptions, and I'll mention those during the talk. So we kind of take a step back a little bit and think a little bit about label-free assays. People mean somewhat different things when they talk about label-free, depending on where they're coming from. So in a strictly logical sense, a label-free assay would cover a lot of classic assays that don't involve any kind of radioactive or dye labeling of a cell before or during an assay. So there are some very famous biophysical methods on the market now, commercialized, for doing label-free assays like surface plasmon resonance. Biocor, which is now part of GE, is one of the leaders in this area. These are really designed to measure protein-protein interactions, protein-antibody interactions. There's all kinds of other classic technologies like calorimetry that, in a theoretical sense, don't involve labels. But most of the time when people talk about label-free, they're either talking about these biophysical methods, or what I'm going to talk about today is among the cell-based methods. And again, there's a wide array of different technologies out there, looking at metabolomics, multi-electrode arrays for looking at the action potential of cells. But what we're going to focus on today is really a class of well-established, non-invasive, commercially available methods, which is what the impedance biosensors fall into. And these really fall into two different classes. One is an optical class, resonance waveguide grading. Right now that's actually commercialized by Corning. It's the Corning EPIC system. And then what we're going to focus on is our own electrical impedance, which, as I mentioned, was earlier commercialized by Roche, but it's really our invention. As well, Applied Biophysics is another provider of this technology. And I kind of group all these together because even though we're going to focus on our particular impedance biosensor, in terms of the assays and applications, which is the focus of this talk, really these technologies all give very similar kinds of data. They have similar sorts of features and benefits for the researcher. So really a lot of the choices that people make on which one they buy, it just comes down to market segmentation, which is more appropriate for uses in terms of throughput. And so I'll really focus then on the impedance biosensor assay principle, but just take this as an example of one of the ways in which a label-free assay technology can be used to benefit cell biologist research. So all of the instruments that we sell accommodate some sort of a microtiter plate. I've shown here a 96-well plate, but all of the details of the biosensor I'm going to talk about are the same in our lower throughput instrument. So the one that you have here to play with is an 8-well system, an 8-well plate, and it goes up to 384 wells. So I'm using this 96-well plate as an example. If you focus in on the bottom of the well, in each plate, what we have is a pretty dense array of gold microelectrodes, and these are deployed as sort of a disc, linear disc format. And what happens is when you add cells to the system, so all the data I'm going to show, all the assays I'm going to talk about are done with the instrument in a tissue culture incubator. The cells are under their normal growth conditions, under normal CO2 and temperature control. So under those conditions, the cell will adhere very well to the gold, just as well as they will adhere to your plastic labware, or in this case, our gold microelectrodes are actually patterned on a glass surface. So the way the biosensor technology works is cartooned here. So for simplicity, what we've done is we've isolated 2 of these gold disc electrodes from the array. And so you can think of this gap here as being the gap between the 2 rows. And what the technology does is it applies a very low electrical field across that gap. It's on the order of a 20 millivolt potential. It's much below the resting action potential of most cells. But what that allows us to do is essentially measure the current flow between these 2 open surface area of the surface area of the discs. As you add cells, the cells attached to the disc biosensors, wherever they sit down, they don't pass electricity. So the lipid bilayer acts as essentially an insulator under the electrical field conditions that we use. And so what this allows us to do then is to very sensitively measure in real time, under physiological conditions, several aspects of cell biology. So over time, the cells will start to divide. Obviously, as they divide, they spread out. They cover more surface area of the electrode. The current flow is reduced. And what we actually measure is electrical impedance. Again, I'm not an engineer. I don't have an electrophysiology background. So the simple-minded cell biologist way of thinking about this is impedance is the AC, or alternating current, version of resistance. So you can think of them as little resistors. Important point is really that using this workflow where we actually put the growth media in the well first, we measure the background impedance, because different growth medias will have different ionic strengths. Software sets that to 0. We add the cells, and then basically as the cells grow, they spread out, cover more of the electrodes, and we see a gradual increase in the electrical impedance over time. So all of our data is time series data. So a lot of customers are looking at simple proliferation assays. Probably an even larger fraction of our customers are looking at cytotoxicity assays. So you imagine a large number of cells covering electrodes, you add a toxin. Over time, as the cells undergo programmed cell death, typically, you'll actually see a series of events happen. The first thing they do is the cells actually start to loosen up. They become a little bit less adherent, and we can see that as a dip in the impedance. Then they start to undergo some very characteristic morphology changes, membrane blubbing. Eventually, they'll actually completely detach and lift off, and so we see a gradual decrease in the impedance over time in this assay. And then finally, a small but significant minority of our customers actually use this technology to look at receptor signaling. And it turns out that when you add a ligand to, not all, but almost all, I should say definitely a majority of the disease-relevant receptors involved in various kinds of signaling, almost always these can couple to a change that we can measure very productively. Usually it's an attachment or a morphology change. It can be a very subtle change. So in this cartoon we've shown a cell that's a little bit rounded. When you add the ligand, you stimulate the receptor, and as part of its normal program, it often will have an effect on cell morphology or attachment. So here we've cartooned the cell spreading out a little bit, just covering a little bit more surface area of the electrode, and what we see there is a often transient, but a very clear and significant increase in the impedance in a short time frame. So that's kind of the basic cartoon view of how this technology works. And this is always a good time for me to pause and take questions if anyone has any questions about the sort of the basics of the impedance biosensor principle.
You tell the difference. So one, how sensitive is the change in kind of width of the cell that flattens out a little bit? How do you kind of what the sensitivity of that is? It's not an area, but can you change that? And how would you measure that as opposed to like two cell, like a cell dividing? It seems like that would be a similar signal that would how you call it.
Yep, absolutely. So those are two different and great questions. And they are an important thing to understand about this technology. So the questions are, number one, what's the sensitivity? And number two, how do you distinguish, for example, two cells versus a cell becoming fatter? So morphology change from a cell number change, and then I also mentioned adhesion. So for the first question, I'm actually going to skip ahead a little bit to a different slide. So getting to the sensitivity issue, it's very sensitive is the bottom line. And so what I do to explain that is I go back to one of the early publications. This is from the early 90s. These two gentlemen, Jiver and Keyes, are sort of the academic researchers who pioneered impedance biosensors. And their electrode design is a little different than ours. They use kind of an interdigitated design, where ours our patented design is this beads on a string. But to a first approximation, they're very similar in terms of the data that they output. And they did some back-of-the-envelope calculations in this publication and came to the conclusion that the method is capable of detecting the vertical motion of cells on the order of 1 nanometer, much below the resolution of an optical microscope. So from that, I wouldn't necessarily stake the claim that ours measures exactly at 1 nanometer, but it is important, and you raise a good point, to make people aware that when we draw a cartoon like this, where we've got this blob of a cell, and then it spreads out and covers, it might be 20 or 30% more surface area, just guessing, that's actually very misleading. Cells are very dynamic. They're constantly reorganizing their cytoskeleton. If you look at a time-lapse video, they're constantly moving around on the plate. When they divide, when they undergo mitosis, they actually round up, divide, and then spread out again. And so in that context, that's where we really have to understand that this technology can measure very subtle changes on the margins of the edge of the cell. And the other thing to keep in mind, and I actually didn't make this point, with our technology, there's a single electrical connection for each well. So we're averaging the impedance across the entire electrode array. So we're measuring at a population level what's going on average with all these cells. It works out to about between 70 and 80% of the surface area. So that's an important other aspect of this. We're looking at very subtle changes that are averaged over the entire population of the cells. And then the other question, that's a very common one, how do you tell the difference? And so kind of thinking about this in very simple graphical terms, our technology lets people measure cell number cell morphology, and even strength of binding, attachment. And that's the source of all the different kinds of assays people do. So proliferation, cytotoxicity, migration, which I'll talk about, those are essentially cell number assays. The receptor signaling, which I'm going to focus on in the drug discovery part of this talk, are morphology assays. A lot of stem cell labs look at differentiation. That's a morphology assay. But even the strength of binding, the adhesion, is something we can look at. For example, in this cartoon, we've got the cell, and we've showed some of its adhesion receptors. So the number of those that are engaged can change depending on the physiology of the cell. It can lock down tighter, and we can even see very subtle differences in that degree of attachment. So the way we tell the difference, there's several pieces to this answer. The first piece is that these types of physiological changes in cells almost always happen on characteristic timescales. So adhesion changes are really fairly quickly, minutes, sometimes hours. Morphology changes, those can also be fairly rapid, but for example, a differentiation assay, that's going to take many hours or days, and then cell number or proliferation assays, that's often on a even longer time scale. So sometimes just by the kinetics, we can get a good idea of which of these things are going on. The second answer is all of our customers, when they're setting up a new assay, they include controls if they have them, so treatments that affect these differentially. And then finally, it's important to understand that this technology, it can stand alone for a lot of your workhorse assays, but it is a good idea, again, when you're setting up an assay at the beginning, to do some standard measurements, whether it's simply taking the cell out of the system, looking in the microscope, taking an image, measuring the cells, counting the cells, put it back in and you can let it continue running, or doing some kind of more sophisticated biochemical assay. We always encourage people to basically, you can almost like build a standard curve. And with that, you can always relate back the kinetics and the quantity of the impedance value that we output back to a more explicit mechanistic measurement. Having said that, what usually happens is people have that concern at first couple experiments, or maybe with a new cell type or a new biology, they'll do some of that indexing. They'll run with our technology for most of the workhorse, day to day. Maybe the last experiment, if they're going to generate a figure for a paper, they might do, again, some standard assays just to have it ready. But honestly, what we're seeing now, I've been at this company 6 years. When I joined, occasionally reviewers would ask for some of that. It's pretty well established technology now, and so it's getting rarer and rarer that a reviewer even asks for that kind of rigorous proof, but it's an important point. The impedance value, just by itself, does not stand alone. Same thing if you're measuring protein quantities on a plate reader. Those numbers don't mean anything without a standard or reference. So those are two very good questions that always come up, and thank you for asking those. Another thing I always like to make sure I clarify is that we're not measuring the action potential or the electrical potential of the cell. We're simply using this electronic impedance readout to, again, measure these three parameters: morphology, attachment, and cell number. So that's a very weak electrical field. It's only applied transiently. A typical assay, if you're looking at proliferation, you might measure impedance every 15 minutes. So with the exception of a few seconds every 15 minutes, the cells aren't even seeing the electrical field. So it doesn't really affect the biology of the cells. That's another common concern that comes up at this point. This might be a good time to mention that these plates are actually electronic devices. They're manufactured in what looks very similar to a chip fabrication facility in our factory. It's a clean room, and I'll pass around a couple examples. So our original deployment is actually the gold braille sensor array is printed on a gold slide. It's spread on there and a laser bladed off. And so I can pass this around and you can take a look at it. Every 4 wells is shared by a common ground. And so to connect those wells, the electrode array in the wells to the instrument, we've got contact pads here. And the instrument has a very simple spring-loaded pogo pin. And so that's how we form the electrical contact. So I'll pass those around. You guys can take a look at them if you want. The other thing people ask a lot is, is the gold bioavailable? Can I take my used plates and take them down to the pawn shop? And the answer is unfortunately no. We've been collecting our own plates in the hopes that someday we can find a cost-effective way to recycle the gold, but it's such a thin layer, it's a sub-micron layer. There's almost no biomass or gold mass there. It's just not worth the energy it costs to take it off at this point. Another question?
So this biosensor principle cover can you use principle made of the potassium sodium cathode channel
question. So the question is the impedance biosensor sensitive enough to measure things like a calcium or ion channel sensitivity? And the answer is not directly. So in terms of the effects of ions going in or out of the cell, that's not a significant enough change in the impedance that we see to measure with the system. This system is really tuned to measure cells. However, like I mentioned with my cartoon here about receptor signaling, growth factor receptors, GPCRs, and many ion channels do couple to changes in cell morphology or attachment that we can measure very productively with the system. And at the very end, I'll talk a little bit about our special cardio safety technology, which measures essentially a beating cardiomyocyte in a dish. And so those assays almost always are focused on ion channels, but it's through the effect of ion channels on the cell morphology or attachment. It's not directly measuring the ions either inside or outside of the cell. So that's a very good question. All right, so it's really good that you guys have a good, thorough understanding of this sort of principle slide, because basically everything else I'm going to talk about, applications, really everything stems from this, and if you understand this, you can have a pretty clear idea of what it can and can't do for cell biology researchers. I'd already mentioned that this is a fairly well-established technology. Is that really wavy, or am I just... Yeah, I don't know. It doesn't seem to be a problem with my connection here, so I'm not sure what's going on. I hope no one has a seizure. All right, well, I think you can read it okay. This is just to point out, as I already mentioned, that it's actually a fairly well-established technology, and what that means for people who want to deploy the technology is often, if there's an interesting idea of how it could be used for a certain biological application, we can find an example in our publication database. And I already mentioned that the instruments actually sit inside a tissue culture incubator, and so all of the assays I'm going to talk about today were set up with adding cells to one of our devices, basically putting it in the station and maybe at some point taking it out and adding some treatment, or taking it out and doing some imaging, but basically it's a walk-away technology. Everything's done under tissue culture conditions, and the data is collected for you, and then you come back after the weekend or whatever and do your analysis. No additional reagents, no handling. And the main difference for the people who use this technology compared to a typical endpoint assay is sort of diagrammed in this cartoon. And again, this is kind of in the context of drug discovery, which is my background, but it applies to most of the work that people are doing. So most of the experiments, you do a lot of stuff that you can't see what's going on. You add the cells, you do some kind of a treatment, and then at some point later, you start the process of labeling the cells or taking the measurement, and then you get a single time point. So the difference here is that everything's done in real time. So all of our data has on the x-axis time. Sometimes there'll be minutes or hours, but typically it's several days. On the y-axis is something called the cell index, which I'll explain in a minute, but it's essentially directly related to the impedance value. And so what you get is you get basically an understanding of what's going on before you do any treatments. Typically, we'll let the cells grow for 24 hours. It's a built-in QC. You know the cells are happy, healthy, doing exactly what they're supposed to be. And then you do the treatment, and again, every time point after the treatment can tell you something interesting about what's going on with the dynamics or the kinetics of a cell response to some kind of variable. And just an aside, this is the instrument you have here. It's actually a 2x8 well instrument. It's run off a wireless signal with an iPad, but it can do, with one exception that I'll mention, all the basic assays that I'm going to talk about today. So I'm going to use a proliferation assay. I thought I fixed it. A proliferation assay to kind of go through a typical data set. So I already mentioned every graph, it'll be time on the x-axis. The y-axis is a cell index. It's directly related to impedance. It's a scaled value, and that scaling factor, it just depends on the biosensor array. So a large plate or a small plate, we scale them so that roughly all those 600 publications, they're all comparable. So sort of a data standardization approach. And the other part of it is, back in the beginning, our engineers thought a cell biologist like me, 762 ohms is not really a meaningful measurement, but if I go from 1 to 2 to 4 as my cells double, that kind of has more resonance with a simple-minded biologist. But you can think of it as directly related to impedance. What we typically do then, I already mentioned we put the cell growth media in there. Software automatically takes a reading. You set that to 0, so you don't want to worry about differences in different media. Then we'll add the cells. We'll typically wait 30 minutes. That gives the cells time to settle and begin attaching to the biosensor. That ensures a nice, even array of distribution of the cells. Remember, I mentioned that we're averaging the impedance across the electrode array. So if you have anything that causes the cells to kind of move around and maybe move to the edges, like heating of that column of liquid unevenly, you can actually affect the data quality. So we have a 30-minute rest, you get a nice even distribution, and then after that, typically cells, usually they'll have a lag phase. It takes them a while to recover from trypsinization or whatever handling you've done. Then they'll take off growing. So most cells will have a typical exponential growth for the next day or two. And then finally, at some point, they will fill the space. They'll cover the entire surface area of those electrodes. They'll reach a stationary phase. Some tumor cell lines will actually continue growing. They'll actually pile up on each other. And it's important to understand that we're just measuring the ones, obviously, that touch the biosensor. So when that array is full, we get basically usually a flat line. Every cell has its own unique properties in terms of adhesion, in terms of morphology, in terms of proliferation. And so these traces, these time-dependent curves, they're almost like fingerprints. They can be very characteristic of not only the cell, but how healthy the cell is. Passage number, you switch serum lots, and again, this kind of gets back to the sensitivity. You can actually see the effects on the cells. And it's always important to remind people when they're using this technology, the differences you're seeing with this biosensor aren't caused by the biosensor. They always happen when you do things like passage your cells too long or mistreat them or change serum lots, just that you can see it now, whereas you couldn't see it before when you're only taking snapshots. So another kind of typical assay is an adhesion assay. So it's important to remember that those proteins that allow cells to adhere, whether it's to glass or plastic or to an extracellular matrix protein like collagen, are important not only in sticking, but they're signaling molecules. They influence physiology, including the spreading of the cells. So this is a very simple adhesion experiment. We pre-treated the biosensors with different concentrations of collagen, and we're simply watching the cells as they attach and spread. So the nice thing about the time series data is you can take a slope or a rate of change, or you can measure after it levels out the degree of spreading, all in a simple experiment, and it's done after 2 hours. So here we actually took the plate off and did some imaging, and you can see that you get a good correlate in terms of cell attachment, number of attached cells, as well as the spreading. So a lot of people will ask, how does it compare to a standard assay? So now I'm going to talk just 1 or 2 slides about cytotoxicity assays. Those are very commonly done on this technology. And of course, that's one of the first things our engineers and product development guys did. So in this case, we're looking at input number of cells versus our readout, which is the cell index or the impedance. And then this is a standard plate-based assay, MTT, but you would get the same result with any of the different cytotoxic assays out on the market. So you get a very good correlation in all cases. This is a snapshot taken, I think, at 24 hours, if I remember correctly. Good dynamic range. If anything, we might even be more sensitive on the low end to lower numbers of cells than the standard plate-based assay. But the point here isn't really to say that we're better. We're at least as good in terms of sensitivity dynamic range. So then the question is, why do people want to use this sophisticated technology, which obviously the electronic device costs a lot more than the simple plastic plate that you use for this assay? And the value of that really comes from the time-dependent series. So this slide shows the principle that the time-dependent profile actually can reflect mechanism of action of a treatment. So this is a cytotoxicity assay. These are a lung cancer cell line. In black is an untreated control. It shows that typical proliferation curve. They flatten out after they reach confluence. They cover the entire surface area. And then what we've done is we've treated with different known cytotoxic agents. Some of these are actually clinical chemotherapeutics, or used to be like 5-FU, DOX, MG132, staurosporine, a pan-kinase inhibitor, a tool compound. And what you can see is all of these treatments give you, at the end of 40 hours, nearly 100% killing of this lung cancer cell line. That's what they do. That's why they were developed. But you can see the path to getting there is completely different. So this is a good example in light blue, 5-FU. It is involved in DNA metabolism. Essentially, it's a DNA synthesis drug. So obviously, it takes a while for the effects on DNA synthesis to be manifest in the actual cell killing. And you see characteristically a bell-shaped curve. Another very predictive curve is the PAC, or paclitaxel. It's an antimitotic. I mentioned earlier that when cells divide, they round up. They divide, they spread out. What happens in this experiment is you treat with the mitosis inhibitor. You inhibit cell division. The cells are in different phases of the cell cycle. They rest in that rounded up phase. So this initial dip here over the first few hours is as the cells accumulate in their rounded up phase. Very characteristic, though, of this kind of mechanism is most tumor cell lines, they lack the appropriate checkpoints to make sure everything's fixed. What they'll do is eventually they'll go ahead and try to divide. They'll missegregate their chromosomes. They'll start to die. But what we typically see is a little recovery. So that's coming on 20, 30 hours. Just this little starts to recover, and that's where they spread out and flatten out again. Even though they're on the path to death, they actually slightly increase our impedance readout. And those are two of the very characteristic kinetic profiles that have been established in several publications. So that's not true only for cytotoxicity. Lots of other areas of biology, our customers kind of map out their characteristic profiles and can actually use them to distinguish different mechanisms. One other thing to mention here, when a cell biologist converts from doing typically an endpoint assay where they just have a point-to-point comparison to doing real-time, the issue of data analysis always comes up. So we've tried to incorporate a lot of time series analysis features in the software. This is an example of one that's on the machine that you have here. It's timed to half max inhibition. So the idea is software will pick up the max and the min, and it'll take the x-axis or the time to get to 50% of the final killing. So it's just one way to characterize these curves in a way that can allow you to sort of rank them based on the kinetic profile. So now I'm going to switch gears a little bit and talk about just a couple slides on the specialized migration application. So this is a fairly unique feature to our technology. The other label-free technologies have variants out there that are capable of doing something like this. We're the only ones who deploy it in exactly this manner. And what we do is we use a very classical design, which is a transwell system or a Boyden chamber. So in these systems, you have a two-chamber device, top and a bottom. You add a chemoattractant to the growth media in the bottom. You add the cells to the top. You've got a porous membrane that separates the two. The pores are large enough for the cells to fit through. They sense a gradient emanating from the bottom of a chemoattractant signal, and they'll actually migrate down that gradient. And so what we do, which is unique to our technologies, we print our electrode biosensors on the bottom of that porous membrane. So as the cells crawl through there, they appear on that biosensor, they cover surface area, and we see an impedance. So it's a way for us to measure cell migration in real time without having to do any kind of staining, counting cells, and so forth. And so that's in contrast to the standard assays where you either do a scratch assay, so-called wound healing assay, where you remove cells and watch them migrate in, or a transfer or a Boyden chamber assay where it's the same format as ours, a two-chamber system, but you've got a lot of extra work to do at the end. You've got to do some labeling, some fixing, staining. A lot of labs still will sit there at the microscope and manually count the cells, or now there's automated imaging. So what we do basically is we set this up and you walk away, and the data is collected for you, and you've got a real-time readout, potentially hundreds of time points and kinetic data to work with. So some of that data is shown here. These are some of the very early studies that we did with very common cancer cell lines in our lab, HD1080 and HeLa. Red and green are negative control, so there's no chemoattractant in the bottom. There's no reason for them to migrate. And then in yellow and blue are two different cell densities where you've got a chemoattractant gradient. The cells sense it. They migrate down, attached to our electrodes. A couple things to notice here. It's a very characteristic profile. Again, different cell lines have very different kinetics. And the other point here, of course, is just comparing either to the input cell number, or you can actually, at the end of this experiment, take our device apart and do the fixing and staining and counting just like you can with a standard assay. And so when we do that, we always get a very good correlation. So just a few examples of more like real-world applications. So this is mimicking what someone who's interested in possibly a cancer chemotherapeutic agent, which would inhibit migration, would do. So locostatin's like a tool compound. It's an inhibitor of a kinase involved in migration. And so in this experiment, negative control is in green. There's no chemoattractant. SFM is serum-free medium. In this brownish color, we've got the positive control, 10% FBS. It's got all the growth factors in cow serum. In the bottom chamber, you see a nice migration. And then what we've done here is we've titrated in increasing amounts of the inhibitor. And you see clear inhibition, both a decrease in the rate of migration as well as in the final number of migrated cells at the end of the experiment. So the big difference here between what people normally do is people would normally do this experiment, set it up, and they'd do a time slice. So they'd take one time point on a counting assay. And what's nice about our system is you can basically do that. You can take a time slice through any part of these curves and generate a dose-response curve. You can generate a potency or an IC50 value for the inhibitor. And it all ends up being pretty significant differences depending on which time point you choose. And everyone who does these kinds of assays knows that. But what they don't always think about is you're compromising. By picking a single time point, you're always compromising on getting the full picture. Relevant to some of the vascular work going on here, people use this to study vascular cell migration. Here we've just used standard HUVEC cells. We're looking at migration towards the basal media. And again, you get a dose-dependent response with good sensitivity in terms of the efficiency of migration towards the chemoattractant. And just finally, there's some recent published work where one of our customers is looking at tubulin acetylation, which has been correlated with aggressiveness or metastatic behavior of breast cancers. And they can see, again, in a time course, a really good migration with normal tubulin. And then they've got a mutant here that's not capable of being acetylated, so they've reduced the acetylation. And you can see a significant reduction in the kinetics. And again, they did exactly what I always recommend. It was just at the end, take it apart, fix it, stain it, and verify in a normal microscopy image that this difference is due to a difference in the cell number. So I think you can see a difference in the density of the cells there. All right, so now I'm going to switch and for the last few slides just talk about some of my own work or the work that I was involved with our internal drug screening team. So before I do that, does anyone have any questions kind of about the basic applications of this technology? Okay, good. So just briefly, this is a kind of an old-fashioned, I think, now cartoon about the linear model for drug discovery. It's a little bit misleading in that no one does things exactly this simply anymore. There's a lot of more sophistication than there used to be in the drug discovery process. Nevertheless, all these steps have to be covered, and so it's important to point out that our technology can be deployed and can actually accelerate the drug discovery process in many phases. I'm not going to talk today about target validation, but people do a lot of siRNA, shRNA assays in our technology. Sometimes you can do the transfection right in our plates and look in real time. I'll talk a little bit about some of our own work, assay development and primary screening on our oncology targets. I'll show you one example of doing some follow-up screening and some lead ID, and then at the very end I'll talk a little bit about preclinical cardio safety. Okay, so I just outlined that here. So some of the work I'm going to talk about actually goes on in our high throughput system. So it's a 384 well system. It's the opposite end of the spectrum from the system you have here. Just 2 points to make about it. One is it's designed, to be integrated with robotics. So we've deployed it on a Beckman FX system. We've got 4 of these stations, and the robots handle all the moving the plates around, dispensing the compounds. And so basically we set it up and walk away, and typically we'll monitor on the order of 20 or 30 plates simultaneously. So a lot of the data I'm going to show you was done on this system. The other point, the system's a little bit unique in that it sits out in the open environment. So for the time it's being read, it's actually not under CO2 control. We do have temperature control. So the way the system works is we put the system on there, the plate on there. We'll monitor those morphology or attachment changes in real time right after we add a test compound. Might be minutes or maybe an hour, and then we'll take it off, and then the machine the robots just basically shuttle it in and out of the incubator. So it might be out of the incubator for just a few minutes when we read for the long-term viability assay. So slightly different than the regular system, which just sits in the incubator all the time. So the approach we took to our own drug discovery work kind of falls in line with the concept of oncogene addiction. So it's the dependence of cancer cells on a single oncogenic pathway or protein for sustained proliferation or survival. These are very well-established targets. Nothing really novel here. Lots of big pharma are working on these same targets. And the way it works is that basically a lot of tumor cells are absolutely dependent on hyperactive or constitutive signaling through these molecules. And so if you inhibit them, in many cases you can find a very effective way to basically kill the cancer cells. So what this looks like in our system is if we take a tumor cell line that's not oncogene addicted, so for the particular receptor we're going to use as our test, we add a growth factor to the receptor, and we see basically nothing happens. In contrast, other cell lines where their wiring or circuitry is such that they've got constitutive expression of the receptor, they're really ramped it up. If we add an inhibitor at this time point, we block that constitutive signal. And what we often see is we'll see a dose-dependent immediate change. That's, as I mentioned earlier, usually indicative of a morphology or an attachment change. And it comes from the fact that I had mentioned in the very beginning that almost all the disease-relevant receptors that people studies, GPCRs, ion channels, in this case kinases, do couple to changes in morphology or attachment. So we can actually use that as a signal, a biomarker for what ultimately you're interested in, of course, is you want to kill the cells. So at the long term, the cells die at the higher concentrations. But what we do is we actually use this as an indicator, a biomarker. It allows us to develop really rapid assays, and we can quickly move with the morphology assay through a lot of compounds. So here's one of the actual cell lines we used. This is a RTK, receptor tyrosine kinase, hyperactive cell line. It's in one of these addicted lines. So in our tests, when we had the test compound, this is actually a compound in the clinic. I believe it's a Pfizer compound. You see it in a short time frame, a dose-dependent rapid rise in the impedance readout. And so under these conditions, we're focusing mostly on that initial morphology response. If you go out a couple of days later, the cells are going to die, but we don't need to worry about that now. So what we did is we actually ran a small molecule library. Total in this case was about 30,000 compounds. So these are a variety of small molecules that have been designed to inhibit kinases as a class. And so we run them in 384 format. Here's some raw data. We always have that control, that Pfizer compound on there. Anything above the noise here is considered a potential drug candidate, a hit. And so if we look at all the data and rank the hits, you can see that we've got a decent number out of this 30,000 of possible individual wells that showed at least 5 standard deviations above the mean. And this is just a common statistic in high-throughput screening for the robustness of the assay. It takes into account both signal to background and variance. And anything above 0.5 is considered a good assay. So we're bouncing around a little bit, but this is actually quite good for a cell-based assay. All right, so what we do is we take those hits, and we again run them through the oncogene addiction, but now we can start to panel them against different cell lines, cells that either have the growth factor overactive or ones where we stimulate it by adding, the growth factor receptor overactive, we stimulate it by adding the growth factor. And so that's kind of how we do the validation and secondary assays, again, using this impedance or label-free technology to really drive the program. So here's an example of a normal cell, which has got overexpression of the receptor. We add the inhibitor, nothing happens until then we stimulate with the growth factor, and we can see a dose-dependent block compared to control of the immediate effect of a growth factor. So we're looking at an inhibition here. And this is a different cell line, a glioma tumor cell line, which has got the hyperactive response. And again, what we're looking for here is basically the long-term morphology and cytotoxicity. I described earlier the specialized cell invasion and migration assay. Obviously, the ability of cancer cells to migrate and metastasize is a key therapeutic angle. So we do deploy that later on when we get it down to a reasonable number of test compounds. So here's one example of our control, which shows a dose-dependent decrease in the rate of migration on this top chamber, bottom chamber system. There's one of our candidates. We're looking at micromolar potency. That's actually a fairly potent compound now. And then finally, kind of moving later on in this program, we have chemists who then take some of these compounds and they really work on them, produce a bunch of variants, lead optimization, and from there they were able to generate compounds that were really in the low nanomolar potency. So this is a strength of potency that you can start to think about actually advancing this into preclinical development. So again, this is the morphology assay, looking at a dose-dependent response, and this is a confirmation in a standard phosphorylation specific antibody-based Western blot. So we're looking at inhibition of the receptor that we're targeting. So finally, just the last couple slides, I do want to mention this specialized cardio system, just because it's a very unique, again, both the migration system and then this cardio system, they're very unique to our particular deployment of this impedance technology. Cardio system, what we did is we took the standard 96-well system and we simply reconfigured the electronics to increase the rate of data acquisition. So our normal system, if you really push it, you might be able to get a 5-second interval between different reads, but really more realistically, it's a minute. Typically. With this one, it's a millisecond time scale, so 12 milliseconds. And what that allows us to do then is to take cell models for cardio function. So you can now generate from a pluripotent stem cell, so a biopsy, generate a pluripotent stem cell, redifferentiate it into a cardiomyocyte, and those will actually beat in culture, in the cell culture incubator. You can keep them beating if you treat them well for weeks or even months. And so these are a very relatively new and fairly exciting new way of doing cardio safety testing. A lot of the big drug companies, they waste millions of dollars on testing things in mice and dogs, and then they get into the humans, and humans aren't mice and dogs. And so you have heart attacks in humans, you have to withdraw. Merck had a very famous billion-dollar drug, Vox, that was withdrawn from the market because in rare patients it caused heart attacks. So now we're looking at human cells. Possibly in the future it could even be personalized human cells. And what we're doing is basically monitoring with this impedance readout in real time the beating. So what happens in these experiments, you put the cells on our biosensor, put them in the well, and they're actually fully differentiated. They're not growing. But what they're doing over time, if we monitor on an infrequent time scale, like every minute, is they're actually filling up that well, covering the surface area, spreading, and they're forming syncytia. They're forming electrochemical coupling, really strong gap junctions. And so as that really strong cell-cell contact forms, you can see over time this gradual increase in the impedance or decrease in the current flow. Then what happens is, if you look in the microscope about here, what you'll see is different foci start beating independently of each other. And because we're averaging the impedance across the entire electrode, that signal tends to cancel out. But eventually, as the cell-cell contacts become more robust, they synchronize. And this whole sheet of cells will beat in unison with a very regular pulse. And so that's what we're seeing at the end here. We get a very regular pulse. And what that allows us to do in our own drug discovery program or our customers to do is essentially test drug candidates very early, very cheaply, simply put it in the well with the human beating cardiomyocytes and see what kind of effect it has. And getting back to the earlier questions about ion channels, one of the biggest concerns that drug companies have is very specific ion channels. One of them is the hERG channel, which just by chance a lot of drugs happen to inhibit, and that happens to be the cause of a lot of the toxicity on drugs that end up being withdrawn from the market. So it's a quick way for them to test this in a physiologically relevant system. It's a human cell. It's beating in the tissue culture incubator. So just quick, sorry about that. Quick view of what this looks like, and then I'll wrap it up. Oh, uh-oh. I might not be able to do it. Sorry about that. Yeah, I think I'll have to show just the static images. Sorry about that. It worked at home. All right, there it goes. Please. There it goes. So on the right is a screen grab of our software in real time, and on the left is a video microscopy of these beating cardiomyocytes. So it's a little bit hard to see. The contrast isn't great, but I think you can see these are contracting periodically. You can kind of see it with this cluster here. And then what the machine does is it can measure across the entire 96-well plate if you want to, the real-time readout. It's kind of like an EKG that you would do in a hospital. The difference is we're measuring the contraction, actually the physical change, not the electrical properties or the action potential of the cell. So the way we use that in our own work is basically we look at these inhibitors that we've got, even a control inhibitor, one that's in the clinic being used to test patients, very well known with this class of anti-cancer drugs, they do have cardiac liability. So if you put them in at a high enough concentration, this is probably a 1,000-fold higher concentration than you would see in therapeutic setting, but you do see effects. And so that's sort of our baseline control. And then what we look for is, as we're testing our own compounds, are we seeing things that are in the same ballpark? And so even though this has some toxic effects, it's up at this higher concentration, so this is actually something we would pass. And things that have effects lower on, at lower concentrations, are things we would hold back. And that's how we ended up prioritizing these. So a lot of this work has moved forward. We are working in early clinical now to possibly bring some of these anti-cancer drugs to the market. And the sum of all this part with the drug discovery part is really what this technology, this type of technology does, label-free, real-time, is it lets you accelerate the process. So you can use more physiologically relevant models and use them earlier in the process, and so that you get less false positives out than if you're doing something very artificial, like overexpressing a receptor in a Chinese hamster cell, which is what a lot of classical technologies do, and using a binding assay. This is really looking in the context of the human cell, which is obviously what you want to treat in the clinic. So with that, I'll just end with a list of the extended applications. So this biosensor technology can be used for a lot of different types of assays besides the general ones that I mentioned. I didn't get a time to go into endothelial barrier function, but that's a very common one. I mentioned cardio safety testing, a lot of work with stem cells, but really the ones that we're really unique on are cell invasion and migration, and then at the end this cardio testing. That's really unique to ACEA's technology. Finally, there's a cell-cell interaction. We do have another transwell device that fits in the system you have here, so it allows you to measure the effects of one cell type on another. It's essentially separated by a membrane, just like the migration device I showed you, but the membrane pores are much smaller than a cell, so the cells don't migrate through, but any diffusible growth factor, cytokine, can diffuse through. So it's, again, a way you can look at cell-cell interactions in real time. So with that, I'll just acknowledge that the founders of our company and my boss, especially in the drug discovery area, we had a lot of help from my colleagues in San Diego. We worked together on a lot of this, and we had a big team in our China facility who ran a lot of the robotics assays. So we had a robotic system both in San Diego and in China. And with that, I'll end with directions to our website and inquiries about the technology. You can always email me if you have questions, especially if any of you want to try the machine that's in your cell culture lab. I'm happy to give advice on whether or not an assay would work, and if so, how to go about starting it. So thanks very much for your attention, and I take questions.